Good morning, ladies. When I came in this morning, I realized that what we did on Friday did not completely record. You can't hear the recording well, so I'm in this morning to go over it again with you. This is as far as we got on Friday with notes. We went over an overview of photosynthesis, and then we did a mini lab, which I will also send a picture of the results to you and give you a little description about that. This picture is the overview of photosynthesis, and I was telling them that it sounds kind of crazy, but this is a picture that if you can kind of memorize it in your brain, it will really help you remember photosynthesis. Photosynthesis is in two parts within the chloroplast. That's what this picture shows. Let me get my pen up. Okay, this is happening in the chloroplast. There's a photo portion and a synthesis portion. It happens in two parts. The first part, the light reactions, happen in the thylakoids. And photosynthesis, again, it takes place in two parts. It takes energy to make energy. It takes energy to store energy in the bonds of the carbohydrates that are being made in this process. So the first portion of photosynthesis is just charging these batteries that we have right here. These energy carriers, these electron carriers, are needed to power the Calvin cycle where carbohydrates are actually synthesized. So in the first part of the reaction, okay, all that is happening is the storing of these energy carriers. This is actually the purpose of the light reactions, to make ATP and NADPH, which are energy carriers. And then those energy carriers are going to enter the Calvin cycle where they're used again to synthesize carbohydrates. It's helpful to look at the overall equation, so I'm going to write that for you now. It's CO2 plus H2O yields C6H12O6, that's glucose, plus oxygen. And then we're going to come in and we're going to balance it. Okay, I need 6 carbon here to match the 6 that I have right there. Okay, I need 12 hydrogen on each side, so this is also 6. And then by balancing that, I have thrown my oxygen out of whack. Okay, and I now have not enough oxygen on the product side. So I also need to balance the oxygen with the 6. So this is a balanced equation. This is a summary equation, meaning that photosynthesis doesn't happen in one big reaction in one big step. This is summarizing the steps. And if you look at this diagram, you can see what's coming in and out of each step. So in the first phase, in the light reactions, okay, light comes in and it splits water. Well, light comes in and splits water. And when that water is split, it harvests an electron that is going to help continue something called the electron transport chain that allows us to make the ATP and the NADPH. In the process, when water is split, again, the reason it's being split is to harvest an electron, which is what we're going to talk about today. So that'll make more sense today. But what the plant is doing is it's making the ATP and NADPH, but in the process, oxygen is made. So one big misconception is that the purpose of photosynthesis is to make oxygen, and that's not the case. That's just something that works out well for us. All of life has evolved to use oxygen, or a lot of life has, because of photosynthesis. Okay, so if you're looking at the summary equation, okay, water comes in and oxygen comes out of the light reactions. And if you look up here, notice what's confusing about this is ATP and NADPH. That's the purpose of this reaction, is to make energy carriers, but it's not in the overall equation that we have right here. And the reason it's not is because if you look, it's recycled. Okay, so it goes into the Calvin cycle where it's used to make sugars, and then it returns to the thylakoids as NAD plus and ADP. Okay, these are our electron carriers that are going to be charged again in the light reaction. So one of the tricky things that you have to remember is that the purpose of the light reactions is to make ATP and NADPH, and that's not actually in the overall equation because it never leaves the chloroplast. Another big misconception is that you need light in order to make glucose, and that's not the case. The Calvin cycle is also called the light independent reactions, and if you see there, it's happening in the stroma. I'm going to run out of space. Okay, that says light independent. I'm sorry, it's kind of smushed together. Okay, but in the light independent, CO2, carbon dioxide from the atmosphere, goes into the Calvin cycle to make sugar, to make carbohydrates. Okay, so that's how I remember that. CO2, CCC. CO2 enters the Calvin cycle to make carbohydrates. And it's not directly needing sunlight. What it needs is ATP and NADPH. So the reason that plants require sunlight for photosynthesis is to charge these batteries to make ATP and NADPH, which will power the Calvin cycle. So these are actually the limiting factors of the Calvin cycle, not light. Okay, light is being used to make these. Okay, but the plant will have a store of this, and photosynthesis can continue, for example, through the night or for a few hours before you run out of energy carriers. And then again, that's the limiting factor. So once you run out of ATP and NADPH, then the plant does require light. A plant being kept in a closet or away from the light for an extended period of time, photosynthesis will stop. But the reason it's going to stop is because it runs out of the ATP and the NADPH, which are made in the light reactions. You do not directly need light for carbohydrates. Okay, and that was the purpose of the lab we did on Friday. So I'm going to explain that to you briefly as well. But let's come back to the bottom of our equation. CO2 is the reactant of the light reactions. CO2 is the reactant of the Calvin cycle, the light independent reactions, and the product is glucose. So this is what's happening in the light independent. And again, you need to be able to recognize that as the light independent reactions or as the Calvin cycle. They'll be called either one in practice problems and when you're reading about it. So this is the overall picture of photosynthesis. Water comes in as well as light. Light is the energy to power the process. Okay, light splits water, harvesting electrons. That allows us to make ATP and NADPH, okay, which go into the Calvin cycle and help power the synthesis of sugar, of carbohydrates, okay, using carbon dioxide from the atmosphere. So again, today what we were trying to address with lab is that light is not directly needed to make glucose. Light makes ATP and NADPH, which when it runs out, prevents a plant from doing photosynthesis. So in the mini lab that we did, we took leaves of the geranium plant, and I covered some of the leaves in foil for 12 hours and some of the leaves in foil for 3 hours. Okay, then we took these leaves, we boiled them in water to rupture the chloroplast. This is all in the handout. But we boiled it in water to rupture the chloroplast, and then we boiled it in ethanol under the fume hood to remove the pigment. Okay, so to remove the green. And after we had done that, we brought it over to our lab tables and we tested it for starch using iodine. Remember that starch is a polysaccharide. It's a carbohydrate, so it's made from photosynthesis. When a plant stops doing photosynthesis, it's not able to produce starch anymore. So after we did our procedure, we tested them for starch to determine where photosynthesis was happening. To determine where photosynthesis is happening. And those are the results that I'm going to give to you, and you're going to write a CER based on that experience. So remember, based on that experiment, remember what you need to know to be able to understand those results is that plants do require light, okay, but they don't require light directly to make carbohydrates. The light helps charge the ATP and the NADPH molecules, which allow the Calvin cycle to run and make carbohydrates. So think about when a plant is put in darkness, do you expect it to immediately stop photosynthesis, or do you expect it to be able to continue photosynthesis for a few hours until it runs out of NADPH and ATP? That's the basis of the experiment that I'm sending you guys today. So thank you, and I'll touch base with you guys in a little bit. Message me if you have any questions.